Transcription is the first stage of gene expression process. It is the process of creating an RNA copy of a segment of DNA. The copy of RNA is then used to make protein in the process called translation. So in this topic, we're going to focus on the transcription. But first, let's just recall from our discussion on the structure of nucleotides that the building blocks of RNA, ribonucleic acid, are very similar to those in DNA, deoxyribonucleic acid. So in RNA, the building block consists of nucleotide triphosphate that are composed of ribose sugar instead of deoxyribose sugar, nitrogenous base, and 3-phosphate groups. So the key differences between the building block of DNA and those of RNA are the RNA molecules are composed of nucleotides with ribose sugar. And also RNA utilized uridine, a uracil containing nucleotide, as opposed to thymidine in DNA. So note that uracil and thymine are structurally very similar. So the uracil is just lacking of methyl, which is CH3 functional group compared to thymine. During the synthesis of mRNA from the DNA template, these are the following pairing of DNA and RNA. So, for example, you have the G CAT on the DNA template. So the pairing in the mRNA will be G will pair with C, C will pair with G, A will pair with U, T will pair with A. This is because there is no thymine in mRNA, right? And then thymine is replaced with uracil. So if the template DNA is ATGCAT, the mRNA strand should be UAC GUA. The transcription in prokaryotic cells again is the synthesis of complementary strand of RNA from DNA that codes a gene, right? So there are three kinds or types of RNA. The first one is the messenger RNA, mRNA, ribosomal RNA, which is the rRNA, and the third one is the transfer RNA, which is tRNA. These are actually briefly the functions of each type of the RNA, as well as whether they are translated or not during the translation process. Okay, so the mRNA is the link between a gene and a protein. So this mRNA contains codes for sequence of amino acids in protein. So the function of mRNA is to carry DNA master code to the ribosome. So these mRNA sequences will be translated in the translation process. Okay, so for tRNA, the transfer RNA, it is actually the decoder of the mRNA message during the protein translation. So it is the cloverleaf tRNA to carry the amino acids. The function is to bring amino acids to ribosome during translation. So this tRNA will not be translated during the translation process. The final type is the rRNA. So it is actually the major component of ribosome. So there are several large structural ribosomal RNA molecules. So the function is to form a major part of the ribosome and participates in protein synthesis. So this rRNA also is not translated during the translation process. Here I would like to show the different transcription process in the prokaryotic cells and the eukaryotic cells. So as we know, in bacterial cells, they do not have organized cell nucleus, so they don't have nucleus, right? So in bacteria, the transcription and translation can occur simultaneously. So bacterial chromosome will begin translating an mRNA even before the RNA polymerase has finished transcribing it. Multiple ribosomes attached to an mRNA will form a polyribosome. So many ribosomes are simultaneously working on the mRNA. Whereas in eukaryotes, the transcription and the translation process are separated. So they have nucleus. So the mRNA are synthesized and processed first in the nucleus and then transported to the cytoplasm for translation by the ribosomes. The enzyme that synthesizes the RNA during the transcription process is called RNA polymerase. So the major functions of this RNA polymerase is number one, involved in the formation of initiator complex, which aids in the unwinding process of the double helical structure of DNA. Number two, the function also to synthesize and to elongate the RNA transcript by adding the nucleotide bases, which are either adenine, cytosine, guanine, or uracil. Also, it forms the termination sequences that stop and terminate the transcription. In prokaryotic cells, the entire mechanism of transcription is summarized in 3 stages, which are the initiation, elongation, and termination. At the end of the termination in prokaryotes, the mRNA form is ready for translation. So during the initiation process, the RNA polymerase holoenzyme will bind to the promoter region of the DNA to form a pre-initiation complex. So the promoter sequence here has a characteristic consensus nucleotide sequence, which are highly conserved. Promoter is a region in a gene sequence where RNA polymerase will bind first before transcription occur. So that promoter is a part of the gene sequence, right? So in E. coli, the sequence of a promoter can be up to 40 bases, right? The characteristic of a promoter, the known feature is it contains a Pribnow box. So what is Pribnow box? So Pribnow box, or also known as the Pribnow-Schiller box, is a promoter sequence that contains the TATA-AT sequence of 6 nucleotides. So this TATA box, or we call it as TATA box, is an essential part of a promoter site on DNA for transcription to occur in bacteria. So we have the -10 region. So this Pribnow box is about 8 to 10 bases upstream of the RNA initiation state. So that's why it is called as -10. So you count to the left of the transcription site. To the left of the RNA initiation site. On the second consensus sequence is at the -35 region. So this sequence is actually TTGACA. So we always call it as TT-GACA. So this 35 -35 region helps to direct the transcription process. So this is basically the alignment of several sequences in different genes. So to show you the location and the features, or the location and the different sequences, and the consensus sequence between the different genes. So most of the sequence are conserved except for maybe 1 or 2 nucleotides that are different, but the location is exactly the same. So the Tata box is at the -10. So the centered about 8 to 10 nucleotides to the left of the transcription site. So the transcription site is highlighted in blue color. So the numbering is now for the initiation site is +1, right? So the upstream of the initiation site is minus. So you count it as minus. Right, so you have the -10 region here, Tata Tata TA80 box, and then sequences, and then we have also the TT GACA sequence at the -35 region. So that is on the initiation process, whereby the RNA polymerase will first bind to the promoter region. So the next phase is called elongation phase. So this elongation phase of the RNA synthesis is almost as similar as the replication process, DNA replication. So except that this process use RNA polymerase, but the replication process use DNA polymerase 3. So after the initiation of transcription, the sigma or rho factor will first dissociate from the RNA polymerase. The template strand is read in the 3' to the 5' direction, which means the RNA synthesis takes place in the 5' to the 3' direction. So this is similar to the replication. The other strand of the DNA template is known as the coding strand because the base sequence of the new mRNA is not identical to it, the new mRNA is identical to it, except for the replacement of thymine with uracil base. So instead of A pairing with T, so A is now pairing with U. So the RNA polymerase catalyzes the formation of phosphodiester bond between the adjacent ribonucleotides. So the last stage of the transcription process is called termination. So this termination happens when signaled by a stop sequence known as the terminator sequence. So actually the termination process are either raw dependent or raw independent process. So I'll explain more about the raw dependent termination later. In bacteria, the transcription termination signal code for RNA forming a hairpin structure, which causes the RNA to separate from the RNA transcript, then finally ending the transcription process. So the RNA polymerase then releases the DNA template, which unwinds back to a double helical structure. Depending on the gene being transcribed, there are two kinds of termination signals. One is protein base and the other one is RNA base. Raw dependent termination is controlled by raw protein, which tracks along behind the polymerase on the growing mRNA chain. So near the end of the gene, the polymerase then encounter a run of G nucleotides on the DNA template and it stalls. So as a result, the raw protein will collide with the polymerase, and the interaction with the raw protein will release the mRNA from the transcription bubbles. So that is what happened during the raw independent termination process. To summarize the process of transcription, so this is in short, what are the processes that happen during the transcription. So first, RNA polymerase will bind to the DNA site called promoter region. Only one of the two DNA strands will serve as a template for RNA synthesis for a given gene. RNA is also synthesized in the 5' to the 3' direction, just as the replication process. And then free nucleotides will assemble, growing of RNA chain, where RNA polymerase will move along the DNA to synthesize the RNA. And then the RNA synthesis terminates at the terminator site, and the RNA polymerase and the newly formed single-stranded mRNA are released from the DNA. So this figure is showing the transcription process of DNA. So you have the DNA as a template, and then from the transcription process, our mRNA transcript is formed. So the transcription starts from the 5' to the 3', producing a single-stranded mRNA transcript. And so then this mRNA transcript in bacteria, so they are actually in cytoplasm, but for the eukaryote is from the nucleus is transported into the cytoplasm location. So upon the termination of the transcription process, then the process is complete. By the time of the termination occurs, the prokaryote transcript would already have been used to begin synthesis of numerous copy of the encoded protein, because these processes can occur concurrently in bacteria. The transcription and translation process in eukaryotic cells is a little bit different from the process in the prokaryotic cells. So in bacteria, the information in DNA is converted to the mRNA directly, right? But in eukaryotes, however, the product of the transcription is an immature primary transcript, or called as pre-mRNA. So this pre-mRNA cannot be translated. So before it can be translated, they first must be processed in a complex series of steps. The protein coding region of the eukaryotic genes are interacted by non-coding regions. There are segments in the gene are known as non-coding region. So in order to make a functional mRNA, this non-coding region must first be removed. So in the structure of the DNA of eukaryotic cells, there are exon and intron. So exons are the coding regions of the eukaryotic genes that will be part of the final mRNA product. So in this case, exon will stay. All right. On the other hand, there is this intron. So this is the intervening non-coding sequence. So they are not in the final mRNA. So this intron must be taken out from the sequence. So the transcription of the eukaryotic genes by the RNA polymerase will generate a primary RNA transcript that contains the exons and introns. So this is the pre-mRNA, the immature RNA transcript. So in this process, introns are removed by the process called splicing. So you can see here, this is a DNA sequence in or gene sequence in the eukaryotic DNA. Alright, it contains exon. Exons are the coding region, right? And in between there are actually the intron. So it is not labeled here, but the one in the middle, not highlighted, are the non-coding region. So during the RNA synthesis, so these exon and intron will be transcribed. So these exon then will remain in the RNA, and then process splicing will be performed, whereby all the intron will be removed, so retaining all the exons or the coding region of the gene. Alright, so now you have a mature mRNA sequence then that can be translated into the respective proteins. So finally, the RNA transcripts will be processed by the addition of the 5' cap and the poly A tail, adenine tail. Right, the 5' cap serves as the recognition signal for the translation machinery. So whereas the poly A tail will extend the life of mRNA by protecting it from degradation process. So otherwise, RNA sequences are very unstable.